Just wanted to say hi, everybody, and once again, thank you for inviting me to share some of my research with your community. I had a fabulous time last year doing it in person at 2023 CrabCon, and I met lots of you guys there, and always impressed with your community, with your dedication, with your ethics, and it's always a great pleasure and honor for me to be involved in your CrabCon event. So just wanted to say that before I got underway here. I talked to Mary a little bit over the past 6 months about this talk, and she said she would like me to talk a little bit about how hermit crabs see their world or experience their world from their multiple senses. And so this was basically her idea, and I had multiple students engaged in research projects that were generating images that I could use for this, and so it turned out quite well. So without further ado, I'll give you a little tour of hermit crabs and how they see their world. So I don't know if any of you have seen this before. This is something called the human sensory homunculus, and basically it's a model of a human with the parts of the body that have the greatest amount of sensory perception in them, exaggerated in size, sometimes also designed on how much of the brain is set aside for those senses. And so here you can see in this human homunculus, large hands for the sense of touch also for the lips and the tongue, taste, and then a smaller nose, ears, and eyes, which are less important, and there's lesser parts of the brain devoted to those sections here. So this is an interesting way to look at how important our senses are to us and which ones are more important over others. And of course, I couldn't find a hermit crab sensory homunculus. Here's a great photo, one of Moa's great photos that she sent me a long time ago. But looking at it, it's already a homunculus because it's already got the parts of its body that are doing all of its sensing being the largest that you see. And I slightly rearranged the ranked list of senses on the left compared to what we were just looking at for the human one here. Touch is still the greatest sense for hermit crabs. All of their limbs, their claws, all of those hairs on all of those limbs, and of course both sets of antennae are important for that. Talk about hearing. You don't see ears on hermit crabs like you do on humans, but they basically can use their entire body and every hair on their body for hearing, and we'll talk about that in a moment. They have a great sense of smell, mostly through their antennules, their inner set of antennae there, and then good sense of sight. We know pretty much nothing about their sense of taste, but they probably can taste, and smell and taste is probably concatenated into one function anyway for these guys. So I thought I'd work my way through some of these major senses and show you some close-ups of what they might be doing with these senses and how they're doing them. The first thing I wanted to remind you about was who hermit crabs are and where they come from. Okay, so here's a lovely phylogenetic tree recently generated by some colleagues of mine with all of the major groups of the Anamura, of which the hermit crabs are a major part of that, and they are most closely related to the true crabs, which are right up the top there. I think I can use my, do I have a cursor? I do have my cursor. Here they are up here. These are the true crabs. So this is your Maryland crab, your Dungeness crab, all those, your fiddler crab. So the Anamura and the Brachyura are each other's closest sister groups. So they are related that way. And the Anamura is also a very diverse group with everything from mole crabs or sand crabs or beach fleas, as sometimes called, king crabs, etc. But I want to point out that there are two sets of hermit crabs on this tree here in two different positions. There is at the top there what are called the right-handed hermit crabs. These are all aquatic. They include the family Figuridae, Polychilidae, Parapiguridae, Haplogastridae, and the Lithoidae, the king crabs, which are in fact, even though they look like real crabs, they are in fact carcinized or crab-like hermit crabs. And then down the bottom of the tree here, connected to a separate branch here, is the Cenobididae, the land hermit crabs, and their closest relatives, the Diogenidae. So this means that the hermit crabs as a group. Oh, I forgot to mention, these are aquatic and terrestrial. Terrestrial cynodontidae, aquatic diogenidee, and they are generally considered to be the left-handed hermit crabs, although some of the diogenids have equal-sized chelae, or approximately equal chelae. So the fact that we have these two different groups on two different parts of the trees means that they are not monophyletic, which means they do not have a common origin as hermit crabs. The entire group, the Anomura, does, but each of these groups is slightly separate, which is interesting in that it means that having an asymmetrical soft abdomen that fits into a gastropod shell has been convergently or independently derived in both of these groups. The alternative is the ancestor of both of these groups, which falls down here, right, lived in a shell and had an asymmetrical abdomen, and all these other groups lost it, which is kind of less believable is the one way you could put that. Less parsimonious is the term we use in phylogenetics. So basically, it's interesting to think that these are not each other's closest relatives, the left and right-handed hermit crabs, which is a kind of cool thing to think about. Also that the Cynodontidae are the only terrestrial hermit crabs, and all the rest of the families are aquatic and marine. There's one known freshwater hermit crab from the Indo-Pacific, but everybody else is aquatic and marine. So that kind of puts us into context here for our group where the land hermit crabs actually fit. So let's compare land and water then, our aquatic ancestors and aquatic cousins with the semi-terrestrial and terrestrial hermit crabs. So just a reminder here, the Earth, 71% of the Earth's surface is water and only 29% land, so there's a lot less land to move on to. Water is really great at buffering temperature changes, and it takes a lot of energy to change the temperature of water in comparison to land. And so, for example, there are greater seasonal and daily temperature changes on land than there is in water, which means it's a more variable environment for crabs to move into. The water, seawater, is already full of all the dissolved minerals that a hermit crab needs, especially the ones it needs to make its shell from. But of course, on land, those minerals are available, but they're in the substrate or they're in the food, and they have to be ingested and not simply absorbed through the gills or through the skin of the hermit crabs. Water has a lot less oxygen per volume than air does. There's about 8 milliliters per liter of salt, of seawater at 35 parts per thousand and at 0 degrees Celsius, and it goes down. The amount of oxygen goes down as you increase the temperature of water. But on land, of course, in air, there is 26 times more oxygen per volume in air than there is in water. So if you move to land, you have more oxygen available to you. You can have a smaller gas exchange surface than you do in water. Water, of course, is denser than air is. It is about 1,000 times denser than air, which means it's more viscous, it's harder to move through, takes more energy. You have an increased surface tension. You have to pull your body through the friction of water, but it also has the added benefit that it provides buoyancy. You can carry around, for example, a heavy shell underwater, and water takes some of that weight, and it's easier to move your body in water. And if you're going to swim, for example, you can push against water, which is different to being in air. Air is less dense and less viscous, but you have the disadvantage of the impact of gravity. So any shell you pick up to carry around is going to be heavier on land than it was and is in water. So there are issues associated with that. And associated with that density is something I'm going to talk about later, which is making and receiving sounds. Sound travels faster and further in water than it does in air, because sound is a function of the density of the medium that it's traveling through. So making sounds and receiving sounds underwater is easier, and you can send them further than on land. And so these crabs that moved from presumably aquatic ancestors to land had to deal with all of these changes. And so just to put that in a little context here. And while we're talking about sound, let me give you just a quick physics lesson about what sound is. Right? Sound is a mechanical wave that moves through an elastic medium and results in us hearing it. That elastic medium is air for us. So when I produce sound, I compress air into that compression wave, which then travels through the medium, moving all the molecules. It expands out into the rarefied or expansion phase, and then it goes back to equilibrium again. And that sound wave enters your ear, tickles the hairs that are inside your ear, and allows you to hear it. Water, because it's denser, is easier to compress, and therefore easier to send sound waves through. And so you can send sound faster and farther through water than you can through air. And so once again, when we're talking about making sounds and hearing for our crabs, it's different in the aquatic setting than it is to the terrestrial setting. So to put things in perspective as well, comparing aquatic hermit crabs to terrestrial hermit crabs, I also want to remind you that terrestrial hermit crabs, land hermit crabs, have an aquatic phase of their life cycle, hatching its eggs into zoea, megalophia, and onto land. And you can see I've put these little symbols here, the water drops and the sun drops here, and I'm going to use them for the rest of the talk. When I'm talking about something that's in an aquatic medium, it's going to have the little water droplet next to it. A megalophia that is still aquatic, but is sort of becoming, turning into a form that can walk out of the water, become a juvenile onto land. I've got as a little bit of both, and then the adult is going to be in sunlight there. And I wanted to, while I talk about these, I wanted to see if I could play this little video here, which is very cool. This is from Corina Hedrick in Australia, who sent me this lovely little video of her megalophia swimming in water here, while I talk a little bit about this. So not only do these hermit crabs have to worry about their ancestral transition from aquatic to terrestrial, but during their life cycle, they have to move from an aquatic environment to a terrestrial environment. And so their aquatic larval stages have to hear in water and deal with all of the issues of sensing in water. And then they have to completely change the way that they sense and hear and smell and taste as they move into the terrestrial medium as an adult. So we have this transition during their lifetime that we have to think about. These are some images I showed you last year when we had our larval, I had my larval talk at CrabCon 2023. These were taken by my past student, Anna Clark, who did all the wonderful SEM work on this. Some nice little images of the zoea and megalophia here. Just to remind you, the zoea are more shrimp-like. They have very large eyes already, much smaller limbs, a few spines on their body for protection from predation. Their antennae proportionally smaller. The antennae and antennules are proportionally smaller compared to the adult. Their eyes are proportionally bigger on their body compared to the adult. And they are covered in more fine hairs and plumosetae for dealing with water, for both swimming in water and for feeling vibrations in water. And we'll talk a little bit about that later. And then as it moves into an aquatic megalophia that's still a swimmer, the front half of the body is starting to look more like a terrestrial hermit crab. It's becoming more heavily carcinized. You can see claws there. You've got stiff little bristles on legs. You've still got comparatively small antennules and antennae here. The eyes are becoming smaller as a proportion of the body size compared to the adult here. But it's kind of getting ready for a transition to a terrestrial existence. So we've got that transition going on there. So here's a little closer look at some of those hairs or setae, or sometimes called sensilla, that are on these both larval and juvenile bodies. We tend not to call them spines. Spines are often, this is a spine, this is a spine. They're often continuations of the carapace of the animal, of the cuticle. They're often stiff. They can be mobile. They can have a mobile base and be able to move, or they can be stiff. That's generally what we call a spine. Oops, sorry, I hit the wrong one there. So what I'm talking about here for the setae and the sensilla, these things here, plumose ones, like these feather-like ones. There's some more here on some of their limbs underneath the body. And then you've got some stiffer setae here on the body, on the butt end of a megalophia. So basically, these are used for swimming in the aquatic phase. They're used for a prey catcher, as Zoea megalopa. They can use these fine feather-like CD to capture phyto and zooplankton to feed on. They bring it to their mouth and then they use their mandibles and maxillae to consume it there. So they basically swim with them and they do prey capture, but they're also using them for 3 sensory functions: mechanosensory, which is to feel the world around them, to feel the vibrations, feel other organisms bumping against them, feel the vibrations that are in the water, to hear sound. That's what mechanosensory is. That's what hearing sound is. You are using hairs as a mechanosensory organ. They can be chemosensory. Not all CT do that. There are specialized CT that are able to actually test the water, look for chemicals, and they're innovated, and they tell the brain what chemicals are present there. And the last sense there is called propriosensory, and that is basically feeling pressure on your body, and also the position that your body is in. We have propriosensors in our body, and they are in our muscles and in our joints, and that allows you, with your eyes closed, to know what position your limbs are in, and whether you're under pressure or whether you're lying down, and all sorts of things like that. Whether you're leaning up against something, that's what propriosensors are used for. And hermit crab larvae and adults have propriosensors too. They're often at the junction between 2 joints on their limb, so that when the 2 joints articulate with one another, those sensory hairs or CT get stimulated, and the crab knows that it has moved that joint in a particular direction. But they can also be internal in the muscles as well, like we have propriosensors. So these sensilla or CT on the outside of both the aquatic larval stages and moving on to the adults as well are capable of doing all these types of functions. Sometimes a CT would have a single function, and sometimes it could have multiple functions. It could be chemosensory and mechanosensory, and it could be a propriosensor and a mechanosensor as well. So different functions there. But the main thing to look at here is the difference between the finer, longer, plumose CD on the left-hand aquatic images there versus the more stiffer hair-like or bristles that are starting to occur on the megalopa body, and you can see them definitely here on the claws and forelimbs here, the brachiopods, the walking legs of this one here. Did it again. And so that's getting ready for the transition to a terrestrial existence, where the sensilla are often concentrated in bunches on the limbs of the crab, and they are primarily mechanosensory and propriosensory in this regard. And so here we have a Cephalatus, I think it is, and you can see that the little bunches of bristles on the limbs here, when you look at them under the electron microscope, are small bundles of 3 and 4 stiff little pointed bristles of different lengths there. And they can all be stimulated by airflow. They can be stimulated by being touched by another crab, or the crab touching itself, an antenna to a leg or a leg to a claw. Those bristles feel all of that, so that's the mechanosensory part of it. The ones that line the joints between the body segments can also be propriosensory, where when a crab moves a joint, it can tell which way it's moved it and how much by which of those bristles have been stimulated. And so we've predominantly lost all of those fine plumose, long sensory hairs that the aquatic larvae had, and we've moved on to bunches of stiff mechanosensory hairs on the body of the adult crab. And this, another great picture of Moas here. Here you can really see just how many sensory bristles a typical crab has. And you can see them all lined up here along the edge of the body segments here. These could very well be propriosensory, the same here. Long ones that point forward and overlap joints here. So anything that touches this crab, anytime this crab moves a limb, those bristles will get stimulated. They're all innervated, and the crab knows what it's doing there. So they can be, as I said, mechanosensory and propriosensory. just this year, an interesting paper came out by a friend of mine, John Faunshall, from the Smithsonian Institution, and I quickly summarized a few points he made here and pulled out one of the graphs he had here. He looked at larval and juvenile Cynobita. He did both coconut crabs, Bogus latro, and the genus Cynobita, the land hermit crabs. And he looked at the types of hairs that were on their body. He used scanning electron microscopy, like the image on the left here, and he measured all the sizes and types of hairs that they have on both the aquatic larvae and the juveniles. And he is a sound scientist. He's interested in sound and how it works in both water and in air. And he looked at how sensitive those hairs were to particular frequencies of sound. And even I didn't understand all of his paper because he went a little technical and did some physics on me. But what I got out of it was the following. That the frequencies which these sensilla, he calls them sensilla, we can call them CP, you can call them hairs or bristles, whatever, they respond to are related to the length of that sensilla. And that's demonstrated a little bit here in the diagram at the top here, even though it doesn't exactly say length on the vertical axis. It basically, it's a physical term relating to length in millimeters as to how it's going to interact with the sound wave. And basically, what you can see here is that the shorter, the hair length in water is responding to lower frequencies of sound. That's the blue ones there. And then in air, you have to reach a certain size to be able to detect that frequency, and they detect higher frequencies in air, starting with the orange bars that are there. So basically, longer sensilla detect lower frequencies, and shorter sensilla detect higher frequencies. And remember how I said that in the larval land hermit crabs, in the zoea, they have those long plumose filamentous setae on their bodies. They are specifically there to detect the low frequencies, right? The cycles that are in the blue region down here in hertz that are in water, while the shorter hairs that are on the adult hermit crabs that we see here are actually there to detect the higher frequencies that are more commonly produced in air. So that's kind of the switch between those types of setae. Not only are they not only are they feeding and swimming with them, but those zoea are using it to detect low frequency sound in water, while on land they lose those plumose setae. They develop more tactile and mechanosensory setae, shorter, stiffer bristles, and they're detecting higher frequency sound that travels in air. They also looked at sound production in Cynobita, and this is in the kilohertz that's mentioned here is a little bit different to what I'm going to show you in a minute here in a different paper from 25 years ago or so. But basically they're saying that these crabs produce sound in the 0.05 kilohertz, so that's a 50 hertz range to 100 hertz range. And they think that they can detect that sound production, and this is on land, up to 0.75 of a meter away. So that's about approximately 3 feet away. So this based on the types of hairs that these juvenile terrestrial hermit crabs have, he thinks they can detect that type of frequency up to nearly 3 feet away from their body, which is interesting when we think about the sounds that these hermit crabs make when they're mating and things like that. He also looked at the larval sensillae, which of course are the longer, thinner, more plumose ones, and they can detect sound that is 2 magnitudes lower in frequency than the juveniles do that are on land. And the principal reason he suggested that these larvae have this low frequency sound detection is that in the ocean, the sound of waves on coastlines and reefs and shores produce low frequency sound. And he thinks that these the larval sensilla are being used to orientate to coastal settings where, of course, these crabs are going to want to settle out as megalopa and make their way onto the land. So they're literally listening for the sound of water on land to be able to orientate to land for when they are switching out of their aquatic phase to go to their terrestrial phase. So an interesting paper that came out recently showing you the importance of some of these tactile sensory hairs, both on larvae and on juveniles. He did not work on any adult Cenobita with the work that he did. He worked on preserved specimens in the museum, and it was all based on scanning electron microscopy and lengths of hairs and how they relate to frequencies that they respond to. So an interesting paper. If you'd like a copy of that, I'm happy to send it to you. You can probably find it on the web somewhere. I can give you that full citation later. Now we mentioned sound production, right? We mentioned sound production, how they can produce sound. I don't know if you're going to be able to hear this. Let me see if I can at least get the video to run. This is one that Mary sent me of two of her crabs doing what they're doing, and I can't hear any sound, so you probably can't hear it anyway. But anyway, they do produce chirping sounds, as you know. There's all sorts of descriptions of how they make these sounds. Initially, it was suggested it was rapping or tapping, and that's since being discredited. As you can see here, there's no wrapping of limbs on shells or shells against shells. Aquatic hermit crabs are known to do that. They're known to tap shells together and to tap their chelae onto shells to make sounds. Then it was also suggested that the stridulating organ, which is of course that ridge of corneous spines that are on the outside of the large left-hand claw that they have, that was being rubbed. That's been basically disproven. No one has got any video evidence of them basically stridulating, which is what a grasshopper or a cricket does, rubbing limbs together to make a sound. There was a suggestion it could have been the gnashing of hard internal body parts like mandibles being gnashed together like teeth, or even some part of their foregut, which is has cuticle around it as well. Very difficult to prove or disprove to be able to actually see that while the crabs are making that sound. And the thought is that something else internal to the crab or internal to the shell is being used to generate that sound. And I went back to a 1990 paper by a couple of Japanese guys who looked at sound production in a couple of species of Cenobita, and they had done some recording, and this is a really nice little graph here showing you the chirping sounds recorded from Purpureus, what frequencies they're at. This is a little bit to what I just showed you from the Thornshall paper. These are showing kilohertz that are from 1 to 5 kilohertz, which is 100. That's 1,000 to 5,000 kilohertz, rather than the 50 to 100 that was suggested previously. But you can see that they're making these small chirping sounds. The scale bar in the right-hand corner is 0.1 of a second. So if you put 10 of those together, then you get about 5 or 6 of these sound events occurring in a second. And so that's what's creating the chirping sound that's there. And their suggestion, based on some experimenting that they're doing as well, that it was the uropod that is used to hold the crab in the shell that was being moved and rubbed against the shell to make the sound. And the uropod, as you probably know, here's a SEM image of the uropod of one of the land hermit crabs, and it's got a scaly rasp-like structure, which is used to actually grip the inside of the shell while they're in there. And if they scrape that against the shell, these authors believe that that's how they're making that chirping sound. They also noticed that crabs in different shells produce slightly different frequencies of sound, and that would make sense. If you have a thin shell, it may reverberate sound differently to a thicker shell. And they actually showed this by cutting holes in the upper part of the shell where the crab's abdomen was going to be and where the uropods were going to be, and watched the abdomen move while they were chirping, and could see the uropod actually moving inside there. And this would be a great thing to look at if you had some of those wonderful glass gastropod shells and you were able to actually see the crab moving while it was chirping. So this sort of sound production is really obvious. It's being reverberated through a shell, probably into the substrate. It's traveling through the air and through the substrate. And of course, the adult crabs are picking up these sounds through all of their mechanosensory setae on their bodies, on their legs, on their antennae that are touching the ground. And they're also up to a certain distance, perhaps up to 3 feet or so, they're picking it up through the air, and then beyond that it would probably be through the substrate. And the harder the substrate, the faster the sound and better the sound would travel. And the more liquid or fluid or sand like it was, it would be the sound travel would be a little slower and would be quite close to the part. And so associated with sound, receiving sound is the ability to produce sound, and we now know that they are doing that definitely in a terrestrial environment. We have no evidence that Zoea or Megalopa are producing any sound in the water, although they very well could be through different means. So that's a little bit about sound, mechanoreception using seeding in both aquatic and terrestrial environments, and about how that sound could be both received and made by juveniles and adult hermit crabs. So moving on to smell or taste, or basically smell and taste possibly. All of you are very familiar with how your hermit crabs use their 2 sets of antennae that they have. The 2 outer antennae, often called antenna number 1, are often waved in the air, or in this case, as you can see in this video from a student of mine, Leah, she sent me this, they're often touching the substrate as well while they're moving. And then there's this second or inner set of antennules, often referred to as antenna number 2, which are often waved frantically in the air, sometimes touching the substrate. They touch other crabs with them as well. And that is constantly testing the air. And each of these antennae is made up of 3 basal segments, and this is a great figure I got from one of the websites, the crab websites that you guys run. And you can see there are 3 basal segments for both the antennae and the antennule, and then they have what's referred to as a flexible flagellum, which is like a whip. The more segments you have, the more flexibility it has to it, and the antenna has a large whip-like flagellum, while the antennule is a little shorter. It has multiple segments on it, but it is a little bent structure and not necessarily a long whip-like structure. And the antennae that are shown in this diagram here, antennae and antennules, are from an aquatic hermit crab, not from a terrestrial hermit crab, which I'm going to get to in just a minute here. I also wanted to take a second to go back, just a second here. This video here, I want you to see that the female on the bottom is actually flicking her antennae back over her shell to feel the male or to communicate with the male that is there. Okay, you can see her doing that. The male's actually not using his at the moment, but you can see her flicking them back, and she's using it to sense behind her or to send messages to the male that is there. So I meant to point that out when that video was going, but I forgot about that. Okay, so let's talk a little bit about antennae. This is the outermost set that are the main, the whip-like antennae. And here we have the Megalopa. This is the image I showed you before. All we can basically see here for the antennae is the 3 basal segments. This is it here, 1 segment, 2 segments, the third one's back there. And there's no real flexible flagellum here yet on a Megalopa. Okay, so they have reasonably short antennules and antennae compared to their body size, and they don't yet have the long flexible flagellum on it. So this is a still aquatic, but soon to be terrestrial individual. And those antennae, they can't be used for much in the way of sensory activity in the water here if they don't have that long flagellum on them. If we move to the adults and look a little bit at them, I've got 2 images of aquatic hermit crabs on the right-hand side here, and you can see a typical antenna is very long, very flexible. The upper right-hand image shows it with brown, black, and white striping on it for the segments that are there, and it has no accessory hairs on it, while the one on the bottom, the aquatic hermit crab there, you can see nearly every segment of the flagellum actually has 1 or 2 long sensory setae coming off it, so it makes it a feather-like structure. It could possibly be used for sensing the water column better, or possibly even catching food items from the water column. And there are some aquatic hermit crabs in the genus Pagurida, which actually live embedded in holes in living corals and never carry around shells, that feed exclusively with feather-like antennae like this, where they're capturing food by swinging it in the water and catching plankton that is in there. But a land hermit crab, the antenna showed on the left-hand side there, a close-up of some of the segments that are on there. And so the statements that are there about mostly being for sensing and possibly for feeding up are both aquatic and terrestrial. The terrestrial hermit crabs, you can see, have a few sensory hairs associated with each segment. There's usually about 4 to 6 that are there of different lengths. Some of them are short, some of them are longer. And this is some of the information that John Forshaw talked about. On those antennae, those different length hairs could respond to different frequencies of sound that are traveling through the air. Now, they also use these antennae to touch objects and individuals and themselves, so these hairs can also be mechanosensory from a touch perspective, as well as feeling sound waves that are traveling through the air with them. So they could use them to hear in air, to hear the substrate by feeling it, and also when they're moving those antennae, they can feel each other and themselves using those sensory hairs that are there. And just about every segment, perhaps except for the very last final segment, has these sensory hairs on them until you hit those 3 basal peduncle segments that are there. Now onto the antennual, which is a lot more interesting. These are all images of aquatic hermit crab antennas. And so the flagellum has been much reduced to a short series of segments that attenuates to a point. I don't know how many is there. There's probably 20 or 30 approximately that are there. And each of them has a series of long setae that are attached to them. You'll notice these are also biramus. They have a secondary, I think this is the, to get this right, I think this is the this might be the endopod and the exopod. One's external, one's internal. And that is missing from the antennae that we just looked at. They do not have 2 limbs on them, they only have a single limb. And so these antennuals have these specialized setae or hairs called astatusks, which is a horrible word, great Scrabble word, always difficult to say. And they are these straw-like setae with blunt ends, which you can't quite see here, but they actually have holes in the ends of them. And these ones here even have little tiny gaping mouths on them, and they are innervated at the base, and basically liquid can go into those holes, and these are used to sense both chemicals on the outside, and the liquid can go inside and be sensed. And so you have a series of long hair-like astatusks inside the curve of the flagellum here. You have several of them laid out here on each of these segments that are external to that. And then along the side of it here as well are these pairs of astatusks. So the entire thing is a chemosensory brush, essentially, that these aquatic hermit crabs are moving through the water column constantly. If you've ever watched some videos of aquatic hermit crabs, they're constantly flicking the antennuals, and they're picking up water, a parcel of water, testing it for chemicals, and then they shake it and throw that old water out and pick up fresh water. And they're using these chemosensory astatusks to sniff the water and pick up all of the chemicals that are there, chemicals of food, of predators, of the environment, and chemistry of the water, etc. So they are a very fine-tuned chemical testing device on the head of the hermit crab. And so once again, on the right-hand side here, we've got the antennual of an aquatic hermit crab, but on the left-hand side here is the modified antennual of your land hermit crabs, your Cenobita. And you can see they still have the short endopod here on the side, and then here the exopod has been, it still has a series of segments. You fuse the distal dozen or so of them have been fused together. You still have astatusks running along the side of here, and then that brush of long astatusks here has been reduced to an incredibly short brush that fits inside a groove on the inside of this antennual. It has the same function. This is a sniffing device, but the astatusks have been reduced in size because we're sniffing air now and we're not sniffing water, and so we don't need the long hair-like structures of the astatusks to sniff air. We just need short brush border to allow the air molecules to get inside to sniff with. And so if we have a little closer look at this, here's a pair of antennules from a land hermit crab. Orientate you again. Here is the flagellum. There's the few dozen here. Along this groove here is where the ascetasts are embedded in. This is them from a slightly different angle. You can see them all in there, and this is what they look like close up. They are a series of short, hair-like structures that capture air molecules, hold them in there, and they're all sensory. They're all innervated with nerves. And so this is how the crab is sniffing the air. This is how the male crab picks up those hormones that you saw Tammy's wonderful video showing you. As soon as the female releases those hormones, the males are sniffing them in the air with this, and they all come running to the receptive female for mating. And so they can, you can see when a hermit crab is waving these around. Go back to the video from a few slides back. You see the crab constantly waving them in the air, and they're constantly sniffing the molecules that are there. And we know they can smell food. We know they can smell dead bodies of their conspecifics of the same species, because that means there could be a shell available. We know that they can smell predators, and we know that they can detect the smell of dead mollusks, because that also means they get access to an empty shell, the possibility. So we know they can very sophisticated sniffing devices for sniffing air, not water, and they've been modified to do that by reducing the flagellum and fusing it together and reducing the size of those ascetast setae that are doing the sniffing there. All right, let's move on to eyes for the last part here, compound eyes. Once again, this is a zoea. They have and I showed you this image last year. They have a large compound eye made up of multiple facets. And they're aquatic. This is for looking in water. And each of those facets has an ommatidium underneath it, which is a cylinder of which light enters and gets trapped inside it and ultimately stimulates a nerve at the bottom of it. And so a typical aquatic zoea has a large eye. It's detecting prey and predators in the water column because it is floating around as helpless plankton and needs to track down food and avoid being eaten if it can help. Compound eyes are a little tricky. They're found in all arthropods, insects, and crustaceans, and their relatives. It's often difficult to know what they actually see. Interesting diagram at the bottom there. We have really good clear vision over distance because we can accommodate our eyes. We can move our lens and focus. The lens of a compound eye is actually part of the cuticle and is unable to be moved. So they have a set focus for their eyes. So unless you move the eye, you can't move the lens. And so it's possible that they have slightly, they would have very good vision at a particular distance, set focal length, and then it's probably slightly blurred for the rest of that, a bit like the insect vision down below there. And you're able to see multiple versions of the same thing through adjacent ommatidia, but they're all going to be at slightly different angles. Remember, if I just go back here one slide, each of these is looking at a slightly different angle. So adjacent ommatidia may see the same thing, but once you move away 2 or 3 ommatidia to the side, the angle changes, and you're going to be looking at a different part of the object. And so you don't get what's called the Hollywood version there, where every single ommatidium is seeing the exact same thing. You see multiples of something in a set area, and then you see slightly different angles as you look away from it. These eyes are specifically designed to detect light and dark really well and movement, because once you get something moves, it moves in multiple ommatidia, and you stimulate multiple nerves, and so you see movement very clearly in multiple places with an eye like this. So the compound eye, the arthropod compound eye, is great for detecting movement. It has limited ability to focus over great lengths and would have a set focal length where they can see best. And unless you can move your eye, which they can on storks to some extent, they would be able to adjust that focal length. We don't know much about what they see color-wise. There's lots of conjecture on the web if you want to look at what they see and what they don't see color-wise. We do know by looking at the genes for the pigments that they have that they can see some color, and some crustaceans see way better than others. And they even see wavelengths that we don't see in some types of arthropods, crustaceans, and insects over others. It's not a uniform type of vision, essentially. And just to remind you how a compound eye is built very briefly here. So as I said, the cornea around the outside is where the lens is. That's part of the cuticle of the crab itself. It's shed along with the cuticle, and it has a set lens that does not move and does not accommodate. And then light gets trapped inside here, goes down through a pigmented column here, basically bounces down through here until it stimulates a nerve at the bottom, and we've got pigments here that react to the light. And so each ommatidium is able to interact with light waves that come into it. And then they're on a domed or concave structure, a convex structure, which allows them to pick up light from multiple different angles here. And there are different ways that this eye, a compound eye, is built. It can be made up like an apposition eye, where every single ommatidium has its own connection to optic nerves, which ultimately go to the brain, or they can be set up where multiple cornea receive light, and then they bend or reflect or refract light down to a receptor axon at the bottom here. So multiple lenses are interacting with one axon versus one lens, one's axon. And so there's different types of eyes found throughout the crustacea. The land hermit are said to have an apposition eye, which is the model here in the bottom right here, although some of their relatives have different types of eyes. So very quickly here, this little phylogeny tree on the left is all of the crustacea. The hermit crabs are in the decapoda up here, along with the lobsters, crayfish, shrimps, and crabs, and they have the greatest diversity of compound eye structure. You can see that, again, I keep sliding my finger on my cursor here and touching the wrong thing. You can see that you have no eyes and very simple eyes in many parts of the crustacea. You have the apposition eye spread across it. It's also present here in the decapods, but you also have several eye types in the decapods that are not present anywhere else in the crustacea, including reflecting, refracting, and parabolic, which is interesting. The light actually passes through the lenses, gets bounced back, and received in the outside of the eye rather than the inside, which is an interesting model. It's a very simplified way how to put that. But you can see hermit crabs are part of the group that have great diversity of eye structure, but they're able to see great movement. They can see color. They're probably seeing things except at a set focal length in a slightly fuzzy fashion. Here's some aquatic crab compound eyes that one of my students, Natalia Rozo, did recently for a project. These are two different species. I actually don't know which ones they are. I didn't look up what hermit crab 3 and 4 actually were for this talk, but you can see all these multiple ommatidia here. These two are the same eye from a slightly different angle, and you can see there's even an area here that doesn't have facets on it, and that could be, I think that's the dorsal part of the eye, and this is the right-hand side is the ventral or bottom part of the eye here. And then this one is another species. You can see that at the edge of the facets are around here. So it has a dome-like set of ommatidia on top of an eye stalk here. So this is a lot of aquatic hermit crabs have this type of eye with lots of facets. As we just saw from some of the great videos we were looking at today, land hermit crabs have a slightly different design on the eye here. Their eye stalks tend to be quite flattened in some species, and the cornea and the ommatidia are concentrated more on the ventral side, the bottom side of the eye, not so much on the top, and to the outside of the eye stalk, and not so much on the inside, which makes sense if you're looking and walking around in a two-dimensional world. You want to see in front of you, to the side of you, and below you to the ground where your food is, and you're not necessarily looking up too much because often your shell is occluding your vision upwards and backwards. And we can see two slightly different eye designs here. Some species, as you well know, have more vertical eye stalks and vertical ommatidia, and some of them have a little more globular or circular spherical ones. And these are both some of Nora's wonderful pictures that she sent me a few years ago, and they're her fabulous portraits she takes of her crabs here. So recently, an interesting paper just came out, in fact, only a week or so ago, by two guys, Elliot Steele and Mark Lavery,
And they've been comparing aquatic and marine hermit crab eyes, and they actually did 2 species of Cynodina, Clypeatus and Compressus here. And they looked at eye size, volume, number of facets, and where they were concentrated on the eye, which is quite interesting. And they said overall, eye dimensions, not the volume, differed between aquatic and terrestrial species. There's no great increase or decrease in volume when you move from aquatic to a terrestrial environment, but the dimensions of the eye did change slightly. They found that C. compressus has a slightly larger eye volume than C. clypeatus, and they said it's because compressus is a more social hermit crab than clypeatus is. I'm not sure where they get that data from. And when they looked at all 3 species, the aquatic crab and the 2 terrestrial ones, they found that there was a higher facet density ventrally or on the underside of the eye compared to the top part of the eye, which goes well with what I was just saying about them really needing to be able to see down and in front and to the side of them and not necessarily up a lot. And I just threw the graphs in there to show you some of the differences that are there. You can see, for example, the upper one here has the number of facets, number of ommatidia that are there with the density. As you can see, the dorsal regions are less in all of them, with the ventral regions all being greater. And then here, the diameter of those facets, how big those ommatidia are, which is interesting, is that the dorsal region, even though it has less facets, has larger diameter ommatidia on it than the ventral side does, which maybe is compensating for the fact that there's not so many there. But in a very interesting recent research article about how hermit crabs see and how you compare marine and terrestrial hermit crabs with each other. And lastly, in the last few minutes, I want to finish up by showing you a few pictures of a very famous hermit crab, one called Winky. Now, Winky was sent to me by, who sent me that? Amanda Mitchell, I believe it was, sent me Winky, but she apparently got it from Mary, and Winky was basically born or assumed his juvenile form with only one eyeball, and only ever had one eye for the rest of his adult life. He unfortunately passed away. Winky was donated to me, and I promised that I would have a look at Winky's eyes to see what was wrong with the eye. Winky. And as you can see, Winky has a perfectly functional right eye stalk with a nice cornea on the end of it. We looked really closely at that under the electron microscope. We could not see any visible facets there, which is quite interesting. It probably means that the facets might be underneath a smooth cuticle that's there, a smooth cornea, and that the facets are actually underneath it. And so when you look under a scanning electron microscope, you can only see the outside. But we looked very closely and could see no facets there, which would say they must be there, but they're just not visible under SEM. And if you notice the other eye stalk is a perfectly functional eye stalk till you get to where the cornea should be, which is right here. These hairs here are these ones here, and it's totally missing a functional set of ommatidia on its left-hand eye stalk, but it has a perfectly well-formed eye stalk that is there. And we went in a little closer and had a look at this, and so you can see this region here is where that ommatidium or cornea should be, and here's the functional cornea. And as I said, this is the edge of the cornea here, around that point there, and you can see you can't really see any ommatidia there, but this is a fine functioning cornea there, and he lived a very good and long life with just one eye. So that was fun to have a little look at the famous Winky that some of you may know of. All right, that's wrapping up my time here. Very quickly, I wanted to make some acknowledgements to all of the American University folks. Andrea Brothers runs our electron microscopy unit here and has trained and helped train all of the students who produce the images you saw today. Anna Cluck, Wade Moldenhauer, Natalia Rosso, Michael Glinos, Clarissa Branson, Leah Estes, all either took SEM images or sent me videos of stuff. And then, of course, also images and videos came from Mary, and specimens, Stacey Boltz, Karina Hedrick, Noel Lundberg, Amanda Mitchell, all sent specimens for use. And I also want to acknowledge several others who have sent me specimens that I haven't used yet. And I know how precious these animals animals are to you, and I thank you for giving them into my care so that I can use them for research. Dan and Samantha Comos have sent me stuff, Shirley Miller and Alison Brandt will have as well. And I think in the future I will get to your deceased friends, and we will see some images of those in the future. So I'm going to get out of this now, and I'm happy to take questions, and I'll go back to a format where I can see myself. So thank you very much. And that, and there I am. Okay. Oh my God, there are so many things in the chat. How am I going to keep up with this?
Stop sharing.
Oh, I have to stop sharing. Okay, I can do that. Where do I do that?
Down to the open share menu. It should give,
There we go. Got it. Thank you. All I need is a few pointers like that. That works just fine. Okay. Wow.
Oh, that was so awesome. That was incredible. Thank you. Just so many things came together. All the people, all the, input, all of your knowledge, all your amazing people that got those images. It's just incredible.
Well, once again, I wouldn't have them without you entrusting your wonderful deceased pets to me. I treat them with respect, and they get used for this sort of work. And they have trained a wonderful bunch of undergraduates on microscopy and crab morphology and crab biology. So they continue to be used moving forward here.
I know you do. And we are happy to provide them, and we're very pleased that you're not going to pet stores and buying them and using those, because you could easily do that.
And that's what I wanted to try and avoid. And so I thank the community for supporting that. And as you all know, if those of you have recently deceased individuals, fix them in an alcohol of some sort, use rubbing alcohol from the CVS store. Contact me, put them in at least 20 times the volume of their body for now, and contact me, and they can sit in that for a long time, and then I'll talk to you about shipping to them. And
awesome.
Be happy to use them. I'll get to them at some point. I've got another batch of undergraduates coming through this next academic year, and so hopefully we'll get something, some new research projects underway with that.
Yeah, like Amanda, you could see your crab used for science and on the screen. I had to leave briefly to help somebody with a sound issue, but I had when you and I talked, I may have missed it, so if I did, forgive me, but we talked a little bit about the crab brain versus the ganglia. Did you cover that at all when I was away, or?
No, I didn't. It's one of the things I forgot. Well, I actually got to a point where I knew I was going to run out of time. And one of the things we talked about was balance, and I decided not to talk about the statoliths and the brain stuff. There's so much I don't know about brains, and there's a lot of literature out there with actually including images of crab brains and all sorts of things like that. I didn't talk too much about that, and I'm not actually doing any research involving brains right now. So I had to leave something out, Mary, and I decided to leave the brain stuff there. I know, just to go back to your questions, the difference between the brain and the ganglion is really kind of academic in many respects. We're often very anthropomorphic about what a brain is, and it's a very large collection of neurons at the head of an organism that controls that organism. As you move down from the vertebrates into the invertebrates, that definition becomes a little grayer, because an invertebrate can have multiple collections of nerves that constitute something we call a ganglion that's in control of certain parts of their body, and not ultimately under the control of any other ganglion. They all work together. And so you don't necessarily have 5 brains in an animal, but you may have 5 bunches of concentrated nerves that are doing different functions in an animal. They often still have a collection that we would call a brain, the one we call that's in the head. Most invertebrates have been cephalized. They have a concentration of nerves in the head because that's where the concentrated sensory organs are: eyes, taste, nose, ears, all that sort of stuff. So we often call the first ganglion the brain. It doesn't have to be any bigger than any other ganglion in the body, and it can have not necessarily have a greater function. So
Yeah.
it's kind of an interesting area. And there are some great papers out there looking at decapod incrustation brain structure.
That's really fascinating to think about too. They can regenerate almost everything. So they're not only regenerating the physical structure, the sensory structure, everything is being regenerated, which is, I don't know, to me, it's kind of mind blowing.
Yeah, oh, definitely so. I mean, those ganglia would have been present in the zoea, but they would have been smaller and doing different functions. You can imagine a zoea with a swimming shrimp tail has an abdominal ganglia that has a greater function than the soft abdomen of an adult, where there's probably still a ganglia in there. It's now doing very different things for what it was doing in a tail, where, for example, when a shrimp has that escape response where it flips its tail,
Yeah.
zoea do that, right?
Yeah.
Adult crabs don't need to do that. Probably the ganglia is in control of that in a zoea. It doesn't have that function in the abdomen of the adult.
Yeah.
So they have a different function there that may be involved in sensory stuff to do with the back part of the crab in the shell and all sorts of things like that. So there's it's a really fascinating area, and I know some colleagues in Germany who are doing a lot of work on crustacean nervous systems and a lot of cool microscopy going on there. I've actually got a student cutting through amphipod and isopod brains right now. Little tiny guys. When they don't have eyes, do they still have the nervous system that comes back to the brain and things like that? There's some of them that live in the ground and in caves don't have external eyes, and that's a protocol that got going with some students now.
Right. That's super interesting. I was wondering too, when you were talking about the CD, and Beth made me think about it. She said eyelashes, we're always looking at those, and from our human perspective, eyelashes. But what's your theory? Is that just protection? If something's coming, you can hide your cornea?
It could well be. Remember, a lot of these guys are nocturnal. They're wandering around in the world today. So if you're going to bump into something, it would be good to know, a couple of seconds before when your ocular peduncle hairs hit something before your cornea does, right?
Right. Saves that.
Yeah. So definitely that sort of function. And it could also be used when they're retracted back in their shell so that they don't scrape their cornea on it every time they go in and out.
Right.
But it's definitely sensory, and it's definitely this constant feedback coming from all of those hairs back into their body and being processed, so they know exactly where every part of their body is when they're in and outside, and what's going on.
Yeah, they definitely know when they're dirty too, because they'll pull them down and clean them off and kind of like wiping their eyes.
Yeah, definitely so.
All right, let me see if there's questions. I know
like a million questions there, Mary. Do you want to be like a moderator and have a
yeah, absolutely. And I will repeat the question so that
yes, please.
Because it's being recorded, but the chat isn't. So, how can we get in touch with you to send the specimens?
Wow. Well, you can definitely find me on the American University website. If you go to the American University website and go look at faculty, look up my name. It has my email address there. The best thing to do is email me. I'm simply ctudge, CTUDGE, @american.edu. Stacy Moult, Stacy Griffiths, Mary, they've all communicated with me, so they all have it too. So you can reach out to somebody in the community as well, and they can probably get you in touch with me. And I'll talk you through getting valuable specimens to my research lab if you'd like to donate them.
So when our crabs are waving their antennae, are they reading chemicals in the air, including smells, or is that only in aquatic hermit crabs?
Okay, we're talking about antennae or antennules. Good question, right? You got two sets there. So the outer set with the long flagellum on them, a lot of the times on the videos I've seen, crabs often have them down, and they're actually touching the substrate with them. Once again, mechanosensory for sure. Especially at night. Think about a crab walking around in the darkness. It's feeling stuff that's in front of it that way. Those inner antennules, less so being used for that mechanosensory function of touching stuff. they're constantly moving like this. And they're in an aquatic crab, they do this and then they go. And they reset themselves. And then they do this and they reset themselves. So what they do is they're sucking water into those acetasks, assessing it, and then they have to get that stale water out and replace it with fresh water. Now, land turtle crabs are doing the same thing. You've seen you guys are doing it either independently or together. They're constantly waving those things. They go down, they come back up. Look at just even the 2-3 little videos I put in there. Constantly look what they're doing. And they're doing the same thing. The air's being pulled into that groove with all those little acetas in there, and the molecules are sticking to the acetas. They're being assessed. And then eventually, I guess, you would fill it all up with molecules and you get rid of and refresh. So that's what they're constantly doing with that. Now, they may use them by touching stuff as well. For example, I've never seen them feed. Do they ever touch their food with their antennas? Is that's a taste thing, right? You guys see them feed? I don't. They're gone by the time I see them, so they never feed. So yeah, they're probably tasting and smelling with that. Less mechanosensory stuff. I don't see so much from all the videos I've looked at them using them to touch things with like they do the longer antennae. So
yeah, that's a good point. I don't think I've ever seen, maybe I have, but not very often, a crab actually spit something out. So maybe they know before they even get it to their mouth from their antennules, whether it's edible or, or something they want to bring up there. I don't think I've ever seen them go, that's what nasty. What's that? You and I have talked before about that one eye and the fact that a lot, who knows, in the wild, maybe they don't live, the one-eyed ones. You told me to check and see if they were always on the same side. And for a long time, it did seem that, but I've since had some on the other side too. I'll tell you my theory, and you can tell me yours if you have one. I think even though they can regenerate something, if they're in between larval stages, they haven't gotten the structure to regenerate, right? So if they lose that eye at stage 4 and then they go to stage 5, there's nothing there to ever regenerate. That's what I'm wondering, but I would love to hear your thoughts.
Yeah, and I think in the past I'd agree with you with that. Remember, the zoea has that great big compound eye, but it's not on a stalk. It's not on a peduncle. It's sitting on the head of the zoea. Yeah. And then that has to transition from 4 to 5 into a stalk megalopa eye that then becomes the juvenile and the adult eye. And I think you're right. If you lose that structure before you've made a peduncle, right, then you're going to completely lose the ability to have that eye. Or during that transition, when you're supposed to be making a stalked eye, if there's some malfunction going on there, whether it's genetic or whether it's physical, then it's going to not have that structure. Have you ever had any that did not have any stalk at all?
Yeah, most of them.
So most of them. They have a single stalk. But this one was Winky was a stalk with no cornea. That's
which is
different. And everything was absolutely perfect until you got to where the cornea should be. All the hairs were in the right place. It was all the same proportion and everything. So that was possibly a genetic thing where when the let's make a cornea genes turned on, something went wrong. They'd already built everything else. That could be slightly different to not having a peduncle or stalk at all, which I agree with you is probably that magic transition from the zoeal stages to megalopus and juvenile.
Yeah, and my ones that never had a second stalk, I would swear that one eye moved just slightly more like a cyclops, over time. I don't know if that's a protection,
Both like this, so I don't know whether it's like physically moved or whether they just hold it over.
It just seemed like the whole structure slowly. Anyway.
Well, it might do molt by molt. I mean, if it's beneficial to them, they could possibly become basically more symmetrical from an asymmetric arrangement. Molt by molt.
Yeah.
Yeah.
Let's see what other questions people have. Please, everybody, come with questions.
I saw Stacey Bolt said she liked the shirt. Thank you, Stacey. I wear it with pride. Thank you.
down. That's what we do. I see Moa there. Thank you, Moa. I love all your pictures. I still want to talk to you. Talk to me. Oh, homunculus. Yes, that's a great word, isn't it? Basically. All right. What are we looking for here? Any more questions there? Left-handed, right-handed. That's an interesting thing. Yeah, it's one of the few, many decapods that have claws, not many of them have that sort of standardized asymmetry in it. For example, if you're a fiddler crab where the males have one big claw or not, the population will have a certain number of males with a big right and a certain number with a big left, and it's not set, but it's really interesting that you have an entire family or super family of crabs where they all have one claw bigger than the rest. And it's such a distinctive picture. It's used by taxonomists all the time. You pick up a crab, you look at it, of course, you're looking at it front on, so it's the crab's right hand or the crab's left hand, and you can say that's got to be in that family. It's pretty standard. So if they lose it and grow it back, it always comes back being the bigger one and the other one. And
Felix Ramono did a wonderful experiment and found out it was related to the shell, how they pull in and cover. When he found crabs and put them in sinistral shells that turn the other way, their other claw got bigger because it was the protective one. He has a bunch of pictures showing that over time. So, it's...
Yeah, but from an evolutionary perspective...
Oh, yeah.
95% of the gastropods in the world are dextral. They're not sinistral.
Yeah.
So you have an entire group of hermit crabs that have a claw that's the wrong size for that.
Right, that's true.
So it's not like you're coming across shells that are 50-50 left or right in the
That's
environment,
true.
right? Sinistral cells are rare. There's just a few species in the world that produce sinistral cells, and so it's a really interesting phenomenon. Not all hermit crabs have the ability to pull themselves in and use that claw like an operculum. Many of them have incredibly long chelae that they just can't ever
That's
get
true.
anyway, especially deep water ones, and there's many that don't live in shells. Right? There's some that live in stones, there's some that live in bamboo, there's some that live in wood, and
Sponges.
sponges and bivalve shells, and they don't have to have claws for any functional thing like that. And yet, if they're in that Pagura branch of the tree, they're all right-handed, and if they're in the other one, they're all left-handed. Or interesting, the ones that are equal are only ever in the Diogenes. You don't find sub-equal claws in the Paguroid lineage, the upper one that we saw. So lots of really interesting evolutionary stuff going on there. It lends great weight to the idea that they independently evolve their asymmetry if you've got that so genetically ingrained into their evolution, where you have the inability to switch, and everybody who's related even has the same size of claws. So, yeah.
Yeah. Maybe you saw this. I saw you say something about Beth. So do you think they have heightened senses with their CD because they cannot hear to make up for the lack of actually hearing, compensating through the hair structures and antennae?
Sensing with hairs is hearing. It's exactly the same thing. They do hear. It's not that they don't hear. They don't hear with an ear like you do. You have exactly the same hairs as a hermit crab does inside your ear, curled up in your cochlea, and if you lay it all out, there's little short ones that get progressively longer and longer and longer ones, and you have a carpet of hairs, and the short ones respond to particular wavelengths, the long ones to other ones, and as the sound rolls across it, you stimulate different lengths, and your brain learns what that sound is. So hearing is sensing vibration. So these crabs are hearing, they just don't have an ear. So you can't separate those two functions. So they are hearing with their antennae. They are hearing with their legs. And if they're feeling the vibrations come through the ground, if the crab's chirping over there and they can't see them, they may still be feeling them. They're still hearing, essentially, that sound through the substrate. So it's the same thing, hearing and sensing. It's mechanosensory. Hearing is a mechanosensory function. And it happens in water as well.
Yeah.
Waves pass through the water, vibrate the animal, all the hairs on the animal. It's heard something, but it's feeling with its body. It doesn't have a designated place to do it, like an ear, like we do.
Do you know roughly when in animal studies we started to drop the anthropocentric model and learn to say, oh, there's all these sensory perceptions that we can't understand in the same way because we don't have them? It just seems like there's been a real upswell lately. I know it's happened.
Yeah, I think different people, some people were enlightened 200 years ago. I think different people and different modes of thinking were more enlightened in the past than now. In general, it's a rare person who still is, or a rare way of thinking that's still anthropomorphic and anthropogenic. We have so much more information available to us now. We understand how things work genetically, physiologically, better than we did 200 years ago. And we understand evolutionary similarities between all of these things as well.
It's kind of like your picture of the elephants mating in the missionary style.
Exactly. I forgot I showed you that.
Christine asks, do you have any students working on crab projects?
I'm actually in between academic years right now. So I just had the last academic year on my summer break, and a lot of those students were in my lab. I actually taught an invertebrate zoology class with a lab, and so Winky, for example, was a class project for 2 students in that invertebrate class. Now, I don't teach that class every year, so I'm not going to be teaching that next year. And I have graduate students coming in, none of them working on crabs. And if I have too many graduate students, I can't supervise too many undergraduates. So I suspect in the coming year, I may only have one student. My wonderful student, Anna Clark, who took all of those larval images, is coming back from study abroad and from summer break and wants to come back to my lab. And I think she's going to be possibly doing a project, but she may be the only undergraduate at the moment. But I got some ideas to do, to look at some stuff. And
awesome.
Yeah.
I'll send you a bunch more specimens this summer. I still have some from last summer in alcohol that I haven't sent. Jesselyn Schultz asks, what's most fascinating to you? I guess she means in the science realm. I don't know.
Oh my gosh, everything. It's hard to say. I'm really interested in everything. I really appreciate it when Mary, people like you come forward and you say something like, you know what, it would be fun if you talked about this. And I suddenly say, oh my God, wouldn't that be fun? It's something I never thought about talking about. Like getting all the sensory stuff together and talking about it, it's not what I was thinking about at the time. And I love it when people do say things like that. Like, have you ever thought about this? And you think, no, I'm going to think about it now. And I can be very easily pushed and pulled in different directions. Crustaceans are always my major love, but I am interested in a very broad range of things, in invertebrate biology in general. I'm a morphologist, so I'm interested in using microscopes to investigate stuff. I'm doing a project with a guy upstairs on nematode worms, right? So we're using the electron microscope to look inside there. That's a cool project because it gets me back to my days of looking inside cells and looking at structures inside cells rather than the whole animal. So I'm really broad interested. There's really whatever's in front of me at the time is the most interesting thing I'm interested in right now. So excellent.
When I hear you talk, it sounds a little bit like you like the hidden things that can't be easily seen. You like discovering the.
I had a morphology upbringing where microscopes were not before I went to college, but from college onwards, microscopes were my main tool, and it's just so amazing to put something that you cannot see with your naked eye under the microscope and be able to say, for example, look inside the cell. Yeah.
And where does it stop? Like we keep thinking we've gone as far as we can go, and then we find something a little deeper and something a little more hidden. And
we've got a microscope in the basement. Well, it's probably not the most expensive one. That if you got the most expensive model of it, you could see individual atoms with it. I mean, that is incredible. I don't know what you do with an image of it. It's possible to go all the way down to the atomic level.
Wow.
you know,
A scent molecule.
we got the cheap scope. It was only $400,000, so we can only get 50,000 times magnification. But yeah, that sort of thing is
Okay. Awesome. Sue Brown asks if it's too far to try and send you samples from Australia because they have variabilis, the species that we don't have even access to.
Alison Brandt sent me stuff from Canada, so I was a little worried about international. International post is always way more difficult than national post is. I'd say it's not too far. It might just be a little trickier with regard to postal rules and stuff. And it's always the case of do not send any liquids, especially liquid alcohol. That's not allowed. So you basically have to fix the individual so that they're not going to rot any further. And then you take them out of the liquid and you send them in a damp cloth, paper, whatever. That's not liquid. So you can honestly say it has no alcohol in it because they don't like that. Once you start sending things internationally, you run into rules about what animals you're allowed and not allowed to. So you may have to put things like this is not a CITES listed species, it's not endangered, it's not blah. I'd say talk to your post office about.
Or maybe her local university, if it was university to university.
All over the world, institution to institution, and we just used to put mailing labels up that said not CITES listed, not endangered, not this and whatever, and it was fine. But now they don't believe people anymore. You know, I don't know really what's going to happen. So it's probably harder. I can't say it's impossible. Once it's fixed, it doesn't matter if it takes 3 weeks or 3 days to get here. I think you will find that postage is exorbitantly expensive for international stuff, and it just may not be worth it. Although I appreciate you sending me your cherished pets, and spending that much money, but I would really look into the worth of it.
Well, and Sue, I think is primarily, they're doing a lot of breeding in Australia right now with variabilis. So I think she was thinking Zoe, but that's even harder to send without any liquid just because
yes,
so small.
Yeah, and it would have to be not alcohol because they're not going to allow you to ship liquid and alcohol.
Right.
So I don't know how you would do that. There may be, you may be able to switch it into a small amount of distilled water if they've been already well fixed in alcohol, and then I'll simply switch them back out again when I get them. That may work. But once again, you're going to be shipping liquid, and they're always a little touchy about that.
Yeah.
But I'll come to Australia sometime. I go every 2 years. I'm quite happy to.
That's right. Save them for him, Sue.
Catch up with those people there, and although being caught at the airport with vials of crabs in your bags is probably the next worst thing to be caught by postal service for shipping them. So
Maybe worse.
Yeah.
Dan asks, what scents do you think hermits are most dependent on? I think that's interesting.
I think that's interesting. And, Dan, it varies by what stage they are and where they are. So when they're a zoea, they're probably feeling the water. They're probably mechanosensory, feeling the water, because that's when their prey is going to turn up. They're going to feel it. That's when a predator is going to turn up. And they've got those great big eyes, so they're probably orientating to light. Those eyes allow them to orientate to the surface and to sunlight and to stop themselves from drifting down into the depths. So they're using their eyesight to position themselves in the water column, but then those eyes aren't good for spotting a prey item at 8 inches away. They would be feeling the water movements of all the plankton that's swimming around them. So they're probably sensing prey, but using those great big eyes to make sure they are staying in the right place in the water column. Because if they sink too low, they're gone. So if they move out of the sunlit zone and there's no prey to eat, there's no predator, no food to eat, then they're gone. So they have to keep themselves orientated to that light. They also need to know when it's day and night, because the nighttime they move closer to the surface, and daytime they move back down again. So as an aquatic zoea, those two functions. Move them onto land, and then I think more than anything, their sense of taste and smell becomes the most important to them, except maybe some sound stuff for breathing. And then if they're moving around at night, their eyesight is pretty much useless. Although if there's good starlight in the tropics and good moonlight, they're probably seeing stuff. But then I think you're shifting to chemosensory function more than anything.
once things get within a few inches of them, they can probably see it, so they're not bumping into stuff, they're not blind, but really they can smell, you know, a dead fish feet away from them more than they can see it, and so it becomes more chemosensory on land. That would be a space that I would well imagine.
We actually use the light reactiveness of the Zoe to our advantage when we're cleaning them or cleaning the water. We'll put a very high, bright flashlight. They'll come to that, and then you can deal with stuff that they're farther away from, or it's easier to collect them and move them because they come to the light.
Yeah,
that's very fortunate.
Common problem for all plankton. You have to keep yourself in the upper 100 meters where the sunlight and the productivity is, because if you drop into the other 9,000 meters below you, you're a goner, right? So they've got to be able to tell which way's up and down and where the light is.
Yeah.
Good question, Dan.
Darcy asks, have you ever considered keeping live hermit crabs to continue your research on behavior, mating, etc.?
No.
Karen weighs in.
I thought about setting them up in the lab here, and then I discovered you guys, and it's like, you know, why would I replicate this when I have such a wealth of knowledge that's available to me? It would take me, how many years of observation would it take me to equal that 20-minute video from Tammy we saw today? I mean, I learned so much in that. I'm a hermit crab reproductive biologist. I learned so much from that 20 minutes than I did in the last 30 years of my life, because I've never seen live crabs doing it. So, you know, to replicate some of this stuff, it would be fun. But it also, I would more likely do it at school than I would do at home. And then I got the problem of, in the summer, I've got no students to look after them, and I'd have to be done. So it's kind of like, I should just lean on you guys, which is what I'm doing.
Yeah, you should. We love it.
You are an incredible community with so much knowledge
and different skills. Like Moa's photographs are amazing. You know, like a little bit of everything you get by having all of us.
Diets, you got people working on safe plants. You got, you know, it's incredible. It's an amazingly diverse community, and you've got so much knowledge there, and you share it so liberally and freely, and it's sending crab husbandry forward in leaps and bounds. It's amazing.
Yeah, we love that.
Can we use husbandry anymore? It sounds so sexist, right? To use another
wifandry?
Yeah. So, yeah.
Worse. Christine says, not a question, but imagine if you could feel something on the ends of your hair. I mean, we kind of can feel when our hair is touched. I mean, is that the same thing? We're feeling it.
You're feeling dead. There's no feeling in the hair itself. It's dead keratin, right? So it's not innervated all the way through. If it was, I think it would be freaky to be able to. I mean, think of the sensory overload you would get when the wind blew, right? Or something like that. Is, you know.
Yeah, or if you could like smell with your hair, yeah, it would be awful.
I mean, I think we're lucky we have so few senses, because we don't have to devote a lot of our brain to dealing with that. But imagine what you'd have to do if your entire body was sensory like that. You would have to be devoting a lot of your brain power to processing and ignoring the stimulus that you get.
Yeah. Do you remember what we talked about? I can't remember exactly what it was, but it was something about energy expenditure. Like why they fall out of trees instead of climbing down, how they make decisions about, I'm trying to remember when we were talking, I thought it was super interesting and I can't recall it now, but maybe it was about the senses, like where do you want to invest your energy as an organism? Where do you expend your energy, you know? Yeah. Oh, I know what it was. We were talking about why they go into their shells and when they go into their shells and falling asleep out of their shell.
Right.
You know, and yeah, if you want to.
You were talking about you can overstimulate a crab to stop it from going back in. Like you touch it once, it pulls in. Yeah, after 6 times, like I'm not going back in there. It's a waste of my time, because you're not dangerous. You're just annoying, and I'm wasting all that energy to keep doing it. So we were talking about something like that. Yeah, I mean that's an evolutionary thing.
thing, right? If you retracted back into your shell every time a wind blew a leaf on the tree above you, right, then you would not have enough energy to perform your regular functions of your day. So there has to be a mechanism there to get you habituated to those sorts of things. Now, eventually, that habituation may kill you, but evolution is driving that. Otherwise, every organism doesn't have enough energy to do everything all at once all the time. So you have to be devoting energy to different things. When it comes to reproduction time, you have to take energy from somewhere else to devote it to reproduction, or store extra energy to devote to it when that time comes.
Right.
And so all those decisions are kind of driven by evolution. And when they don't work, those individuals are removed, and you don't get that reinforcement of that. So yeah, that's kind of all aspects of a crab's life, decisions like that.
And our lives too.
And our life too, that's right. Yeah.
Michelle says, on several occasions, I've seen one of my crabs sitting in a hide, like a coconut hide, and they will hang a leg out of the opening and swing it back and forth before moving out. Curious why they might be doing this.
Wow. It's like they're feeling to see if there's anybody out there before they step out. Makes sense. I don't know why they would use a leg versus an antenna, which makes the most sense. You could just stick that, although you have to stick your head out to put your antenna out, in a sense. I don't know.
I think mine do that, something similar when they're approaching the edge of something, feeling like where their limits are. Can I put a foot down there? Will I get a grip there?
Yeah, maybe the crab's simply feeling where the edge of the opening is, so they know where to go out through, and it looks like they're sticking a leg out. That would make more sense to me than kind of deliberately sticking a leg out. I mean, we don't do that. You don't put your hand around a corner to see this. We look, but then you're sticking the most important part of your body out first, and if there's any danger of being hurt, then you're a goner. So it's a conflict, I guess, that's going on.
If we had 10 legs, we would sacrifice one, and we could regrow it.
Yeah, that's an unusual behavior. I'm not sure whether they're just sensing the opening or whether that really is a deliberate, is there anybody out there before you step in?
Beth Carducci asks, what do you think they make of us? What do they think we are? Once they understand we're not predators and they begin to trust us, I often wonder how they perceive us.
Large, garlic and caffeine smelling, fuzzy balls of noise, I would suspect.
Baby talking. Oh, you're so cute.
Yeah, I mean, and you remember, you only come into focus when you're like so many inches away from them. So you're just like this huge, fuzzy, noise-making ball that smells like caffeine, right? And then all of a sudden you come into focus and they see these humongous eyes and these fingers pointing at them. And it's only that they get used to it that they don't hide in their shells every time you turn up.
There she is again.
Exactly. But they also know, they're also smart enough to know that every time the caffeine-smelling ball turns up, something cool happens. And so they get used to it, right? And so they know if they've been in captivity for 30 years that they're perfectly safe, and anyone that turns up at the glass, is interesting. In nature, that could be different. That would be counterintuitive.
Yeah.
For a wild crab to go running up to something like that would be more likely to be a predator. So
curiosity killed the crab.
Can be habituated when they know better. But as I said, they're not seeing you clearly from feet away. They're hearing you first. They're feeling you walk into a room. They're hearing you walk into a room. The glass is vibrating, the substrate's vibrating, their hairs are vibrating, the air's vibrating. You walk into a room, you sit down. They smell you, your body odor, your coffee, the garlic, the Febreze on your clothes. All of those molecules enter the tank, and they know you're there. And then if you're moving around and backlit, they've seen the light changes, but they really don't see you until you're up against the glass and you're 4 inches from them. I have no idea what their focal length is. Someone may have figured that out, but it has to be set distance where things are in focus. and it's probably a very short distance, probably only a couple of inches between crabs, most likely. Or it's that perfect distance where you need to reach a claw out and grab a piece of food, that's your perfect distance.
Or fight off somebody, yeah.
And so up until that point, you are a fuzzy colored ball that blocks the light and doesn't block it. But
brings them delicious food.
Most likely sound first, smell second, sight after that.
Michaela says, I have a question. My son loves crustaceans and is considering going to college to be a marine biologist or something similar. What do you recommend for getting his foot in the door at an early age? He's 10.
He's 10. Whether you're going to be a marine biologist or not, you need to make sure you get an undergraduate science degree. And it doesn't have to be a marine biology science degree. There are not many institutions in the U.S. that provide a BS in marine biology. There are some big schools, Rosenstiel School in Miami, Rutgers, I think, has one, Field School. But even just a good BS in biology, the concentration is say organismal stuff versus molecular or whatever, would be suitable. Then you would probably concentrate on marine biology in grad school, is the most likely thing, unless you have a good local school with a bachelor's degree that's in marine biology. Get experience, internships in marine biology, field schools, field trips, all those sorts of things. Get that experience even before you head to college, and be curious all the time. And reach out to people who are marine biologists. Ask them how they did it. Everyone's route's slightly different, but and depending on how old they are as well. But there's all different ways to marine biology.
Katie Roberts says, can you sometimes tell why a hermit crab may have passed when you receive the specimens, if a person asks, or are they just for research?
Yeah, I don't have the capability for doing any sort of autopsy type stuff. So generally the answer to that would be no. Once again, you guys probably know better than I do because you watched it get sick, whatever. I remember Dan and Samantha sent me one that they said the abdomen swelled up so much it had to leave its shell. It obviously had some sort of infection in there and then passed. I mean, when I got it, the abdomen was shrunk back down to normal size, and it's like, I really don't know. And you would need to have a pathologist, or someone would have to be doing microbiology or look and see if it's got fungus or whatever. I really don't have those capabilities or that expertise in my lab. So I'm generally not doing that. It would be beyond what I normally do for my research.
Maybe Michaela's son will go into crab autopsy work.
If you ever take your hermit crabs to vets, are vets ever useful to you? Do they ever know anything about something that's not a cat, right? And
Not
so,
really.
or dog. So somebody is going to be useful in the future if they have some information about this. And once again, your community is going to be the ones to gather that information.
Some Virginia Tech veterinary students here this year that came and toured my setup and saw where I raise Zoe and everything, and they were very interested. They were a splinter of exotic pet vets. That's what they wanted to go into. But Angela, the one who lives in the Keys, says we are seeing a lot of big hermit crabs crossing right now, moving more inland. They're doing major road work at the end of the road, pounding and scraping the road. I wonder if the pounding of the road is moving them inland. Your thoughts?
Absolutely. They would be feeling every one of those. I mean, they can feel your footsteps when you walk on the sand. They probably feel you from meters away through the sand. So imagine one of those, vibrating packers that they use. That's probably transmitting hundreds of feet through the ground, and they can feel that. They wouldn't understand what it was, but they would understand it's not good.
Get the heck out of here.
Yeah, I wouldn't be surprised for them to move away from massive sound like that.
Yeah. Christine says, could you freeze dry specimens, or would that ruin the specimen?
It depends on what I wanted to use it for. Freeze drying for something like a crab, it doesn't change it very much because the carapace doesn't shrink. The abdomen would shrink. So, if I was just interested in the external carapace morphology, it wouldn't be a problem. If I wanted to look at the hairs on the soft abdomen, then the abdomen will be all shriveled up after freeze drying for that. If I was going to do like DNA work or something like that, freeze drying would probably work fine. I could just reconstitute it and look at DNA. So it would depend on what I want for the morphology work that I do, and to get those images I got with the SEM, you want to have the crab. It's got to be inside a vacuum. It's in a vacuum and it's coated in metal, and it needs to be in a form that was as close to living as possible, and that's a very tricky process to get it to that. And so you need to start with it as natural as possible. So that's why, in general, a fairly gentle fixative like alcohols and hormones and things are the best things for that. And then you try and maintain it in that shape. Hard things are easier than soft things are to do that with.
So what is the science behind alcohol? Does it replace the water in the specimen?
No, alcohol actually cross-links proteins and denatures them. And denatures them in a state that was in a state that was close to what they were when they were alive. Some of the other fixatives, like the aldehydes, do it much more slowly and are way more gentle and leave things metabolically active more often, but they're all nasty carcinogens and no one wants to work with them. Alcohol is kind of the safest, but the crudest and the fastest fixative we know, and it simply cross-links proteins and denatures them in as close to natural state as possible. There's always some shrinkage, anywhere from 5 to 15%, and then it does exclude some alcohol, some water, but you have to exclude it even further to do the type of microscopy that I'm doing. You have to get even that last little bit of moisture out. You have to move it into chemicals that really don't like water and are aggressively expelling water. And then you basically dehydrated it by putting it in a series of liquids, and then you let it air dry, and then you very gently put it into the microscope.
That's amazing. Ashley asked something that's very interesting. Have you studied, or anyone that you know of studied, whether hermit crabs living in captivity can sense changes in the weather? I've noticed an increase in activity in my Ecuadorians when there's a thunderstorm. I have too. Do you think they can sense the change in barometric pressure, or are they seedy feeling the ground of loud thunder claps? I would love to hear your thoughts on this question.
I'd say probably both. There's a lot of animals that are responsive to changes in air pressure. I mean, some people say they even know when storms are coming in, but generally we're some of the most insensitive organisms on the planet in general. Dogs and cats recognize that. Birds recognize changes in air pressure to avoid storm. So change in air pressure is a definite yes. If they know, for example, if they're terrestrial animals, and they know their dose of fresh water is coming, then a change in air pressure, it really gets them excited if they know it's going to rain. If fresh water is important to them. And then at the same time, a good clap of thunder is going to vibrate substrates, and they will feel that too. But then again, you could test that by, dropping your shoes on the floor, make it feel like thunder and see if they get excited or not. So it could be both or one of those two things, but I'd say definitely changes in barometric pressure.
Or the ions, right? The positive and negative ions, maybe?
Yeah, I mean, you can smell rain coming, and they would be able to do that once it was close enough. We very crudely can smell rain on certain wet surfaces, and we can feel the chemicals that release, smell the chemicals, and they'd be able to detect them even before we can and further away than we can as well. So ions, chemicals, and barometric pressure. And I'm not surprised they get excited. A good fresh rainstorm makes me excited. I like to dance around.
I know. Corinna asks, I've seen the babies all the same size, choosing different size shells. Do you think they have individual tastes? Maybe that's a better answer for a question for us as keepers.
Yeah, absolutely. I watched hermit crabs try 10 shells and choose one of them, and it wasn't the one that I would have thought would have been the right size for it. So I really don't know what they're searching for there. Now there is also internal fit versus external fit, and we look at the shell and say that looks perfect, but we don't know what the inside looks like or feels like. So they could be selecting stuff on the inside that we don't perceive. So that's one thing about it. And if there's no choice, then if the choices are limited, then they're going to take the best they can get at that time. But if it's unlimited choices, then I don't know what they're selecting for, really. It's got to be to do with weight, carrying it around, how easy it is to carry, and all those sorts of things have to come into it. I've heard there's modifying shells by rubbing and increasing entrance size and stuff like that.
Yeah.
They can do that too.
There's at least one scientific article about that uses a hermit crab's shell preference as an argument for sentience.
Oh, interesting.
I quoted it last year in my talk on sentience. I don't remember the authors, but they said, we believe they're sentient because they have shell preferences. They will choose a particular, I mean, it's a form of sentience, but yeah.
Yeah. Once again, we are making decisions about why they chose those shells. We really don't know why they chose them. They might
comfort.
They might taste and smell good. Like this one too recently had a dead slug in it, a mollusk in it. I'm not going to put my butt in that. It smells like that. Give me something cleaner. Or others are like, I love the stinky shell. I'm going straight for it. I have no idea what they're choosing. No idea.
We definitely see ones. We say they like tight pants or they like loose pants, big pants, And
another thing that I only thought about when I read those recent papers about sound production and stuff, the frequency of sound they produce changes according to the type of shell that they're in. So many large volume, thin shells reverberate sound better than the thick shells do. And so that might also, if they are producers of sound for reproduction, a particular shell might be important for sound transmission even. So a big male who's ready for the breeding season might say, I need to get myself a good shell that I can transmit good sound with. Might go up looking for it.
Yeah. Amplify my bass.
Yeah, that's right. Exactly.
Leslie asked, why do you think they spend so much time, energy on their hamster wheels? What is the benefit to them?
I have no idea. I didn't even know hamster wheels existed until last CrabCon 2022. So, I mean, they do travel a lot. They are big travelers in the wild. They are constantly foraging. They're opportunistic feeders. They're literally walking hundreds of meters every night. So it doesn't surprise me that they can be as active in those closed settings. I don't know why wheels are. Why does a mouse like a wheel? I really don't know.
I will say we saw a huge increase in mating and spawning after many people started introducing something where they could travel a long distance. I think it's probably an instinctual thing. I got to get to the ocean. I got to, to have that ability to satisfy that urge, right?
The gravid females do more wheel walking.
Always. I have a video.
The ocean than not. I mean, that's an interesting study to look at.
I have a video of like 6 of my females all trying to get on the wheel, and it was right before spawning.
Yeah, so you keep track of the time, you keep track of the state of the animals, you keep track of how many miles they walk, and you collect that data, and before you know it, you've got good information on that.
One of us, Holly, I believe it is, has a pedometer on her wheel, so she can actually measure how many, how far do they walk in captivity.
As long as she doesn't Bluetooth it to her phone and tell people she did 30,000 miles this week, so that's
right. It was really my crab. But... Let's see.
Yeah, must be getting near the bottom now. I don't even know where we are now. I'm trying to keep up with
the. We may be closing in on it. Let's see.
Oh yeah, there's a question there.
Yeah, I think I'm down to the bottom. If anybody has anything else they want to ask Dr. Tudge that I didn't see.
Questions and you want to pop it again right at the bottom so we can.
Yeah, if we missed it, do it again. Yeah, there's Holly. She keeps tabs.
Okay. Yeah. Oh, interesting. interesting, Darcy, that they have, even in your little enclosures, they have preferences for that.
Preferences. Oh, Katie asked if we think they could see certain colors.
I tried to find a few papers on color vision in decapods. It's really variable and not much work has been done on it. Basically, the best research that's been done is we look at the type of genes they have for the type of pigments they have in their eyes to know what they might be seeing. And not a lot of work's been done across a broad range of crustaceans. Some, for example, the mantis shrimps are the most famous. They can see more colors than we can, and they can see UV and everything. There's a couple of famous ones that some work's been done on. We believe they can see color probably less than we can. But I haven't seen any really good data on exactly what it is that they are seeing.
The other question related is, can they see in the dark?
Yes, they can see in the dark. They would see differently, like we do. We see less color at night because the cones are not as actually. Do they only have black and white? They see black and white. I actually don't know much about the metabolic functioning of their eyes, but yes, they would be able to see. They would see worse, like we do, but they would still be able to see stuff. But they're mostly sensing, using other senses when it's dark.
Can you see Christine's question?
What do you need to get more academic research done? Well, it's always money, you know that. It's always that interest too. You know, there's also a big block about, you know, scientists getting data from places that's not their own. It's something that I've always been open to, and that's why I've run these workshops in the past for you guys. It just has to be, there's data and there's data that's scientifically useful, and you have to collect it in a manner that's going to be useful. So this all started 4 or 5 years ago when somebody told me, we think they changed sex. And I'm like, that's a great question. You've got to prove it. And this is how you got to prove it. You've got to do this, you've got to do that, you've got to do that. You've got to collect the data that convinces somebody from a scientific perspective in a scientific manner to prove some of these things. And I think the biggest problem is a scientist is never going to be able to do as much research as your community can. I can spend the rest of my short time on this planet trying to do research on that hermit, and I'm never going to get anywhere near the amount of data that you guys can generate. You just have to collect it carefully, record it carefully, and make it available. Unfortunately, it's unlikely you guys are going to be able to publish it except amongst yourselves unless you collaborate with somebody scientific, because there's that snobbery in the journals about that. But that your data is good and it should be out there. You just have to collect it so it can be statistically analyzed, or you know, that sort of thing. So you're doing it. I know you're collecting that data. You know, your comments here tell me you're collecting all of this great data. It has to go from anecdotal to being testable, essentially. Yeah. And it's making that shift, and that's a hard shift for somebody who has a full-time job and does this on the weekends. You know what I mean? So I fully understand that. But I'm willing to chat with anybody about stuff that they've got, data that they've got, and we can talk about how you might use it or how you should collect it. So that comment back about forest side, ocean side of your tank. I mean, if you videoed that and had hours and hours and hours of video, and you can statistically analyze how much time they spend on one side versus the other at different times of year, you can probably show this shift. But you couldn't say all 3 of my crabs did that, and yes, that's obvious data and it's obvious what they did. You need to collect it in a different manner to make it acceptable. You know, it's got to be analyzed somehow. That's the kind of the snobbery of science in many respects. So yeah, just keep doing what you guys do and share it amongst yourselves and get it out there as best you can. And there are researchers. I mean, that guy Mark Lavery who did the paper on vision, he's a guy I know. He's up in Massachusetts somewhere, I think, or New England, somewhere there. He's really into this, and he would love to work with anybody. He has a colony of crabs himself, but I'm sure he would love to work with anybody who's got good data on hermit crabs and vision, and he studies behavior and decision making and things like that.
Awesome.
He also does some stuff in the wild as well. You know, to get a study site somewhere in the Caribbean. But yeah, but there's not many scientists who are specifically working on land democrats. A couple in Japan, a couple in the Caribbean, and 1 or 2 in the States, and that's about it.
Well, we are so grateful that you
so-
take us seriously and you bring us this wonderful knowledge and you appreciate us, but we appreciate you so much more. We really do. We are very grateful just to have you as part of our community, as part of our colony.
I feel privileged to be brought into the colony and into the community. It's always fun to interact with you all. Like, as I said, I'm always impressed with your ethics and your morals and everything that you do and the reasons why you're doing it. And I'm always willing to support you in any way I can. So reach out. And I look forward to seeing you all in person at some point again in the future.
Next year, let's do it. Let's make it happen.
All right, anyone, if you want to keep chatting with me, email, I'll answer it.
He will.
All right,
All right.
thank you
And
all.
I'll keep you updated on the breeding this summer.
Yes, thank you, Mary. We'll chat some more about this. And thanks to everybody. Have a great rest of your summer, wherever you are all over the world.
Thank you.
Okay. All right.